Hi guys, Miss Francis here to continue our discussion of Mendelian genetics. Today we're going to focus on Punnett squares. By the end of this video, it's my hope that you know and understand these vocabulary words. So let's jump in. What is a Punnett square? Well, a Punnett square predicts the results of a genetic cross between individuals of a known genotype. Well, what is a genotype? So before we can understand what a genotype is, let's understand what a phenotype is. Phenotype for physical characteristic. In other words, the outward appearance. When discussing peas, we're talking about like the purple versus the white flower or the round versus the wrinkled seed. In humans, for example, do you have brown eyes, blue eyes, green eyes? Do you have blonde hair, red hair, black hair, brown hair? Whereas the genotype is the genetic makeup where we use a special nomenclature to describe an organism's genotype. We use either capital letters or lowercase letters, where the letter itself varies depending on the characteristic that we're talking about. So for example, let's say we're talking about Mendel's flower color, where capital P represents purple because that's the dominant color, and a lowercase p represents white because that's the recessive color. So if I were homozygous or pure dominant, I would have capital P, capital P. If I were homozygous recessive, my genotype would be two lowercase p's. Now let's say I am heterozygous or a hybrid dominant. I've got the genotype capital P, lowercase p. However, my phenotype, I would show the dominant characteristic. So if you have time right now, practice some of the monohybrid crosses. Let's talk about a test cross. A test cross is helpful when an organism shows the dominant trait, but we don't know what genotype it has because it could be a purebred dominant or it could be a heterozygous dominant. So what we do is cross it with an organism that shows the recessive trait. If an organism shows the recessive trait, then it has to have the genotype homozygous recessive, meaning it's got two lowercase numbers. So for example, if we take a black guinea pig, where the black guinea pig is expressing the dominant phenotype, well, it could either be heterozygous dominant or homozygous dominant, where the test cross is going to help us determine that genotype. So let's look at how we use the test cross. Well, the test cross would be the unknown dominant times the known recessive. So here's where we would put the unknown dominant. So the unknown dominant could either be two homozygous dominant, which would be two capital P's, or heterozygous dominant, which could be a capital P and a lowercase p. But the known recessive, in order to show the recessive characteristic, it has to be homozygous recessive, which would mean two lowercase p's. So with our Punnett square, what you do is you put the potential gametes on either side of our Punnett square. So I'd put a capital P, lowercase p. I could put a capital P, lowercase p, and then lowercase p. And it doesn't matter which side, whether you put it on the top or along the side, you put the possible, letters, because it'll end up being the same in the end. So I would end up with a capital P, lowercase p, capital P, lowercase p, capital P, lowercase p, capital P, lowercase p, capital P, lowercase, capital, lowercase. So our F1 genotypes, we've got 100% heterozygous dominant. But on the other example, possibility 2, we've got 50% heterozygous dominant, and then we've got 50% homozygous recessive, or we could simply just say recessive, because the only way it would be recessive is if it is homozygous. So our possible phenotypes, this would be 100% black. This would be 100% black. 50% black and 50% white. So which possibility is more likely? It's more likely that our unknown guinea pig has the genotype for homozygous dominant. This was an example of a test cross that Mendel might have done, where he did not know the purple flower, whether it was homozygous or heterozygous, and he crossed it with the recessive white flower, which had to be homozygous recessive. So what he ended up with, either all purple offspring or half purple, half white offspring, meaning that there's more of a likelihood that the phenotype was homozygous. We explored monohybrid crosses, which focus on the inheritance of one trait. Now we're going to explore dihybrid crosses, which look on the inheritance of two traits. One of the dihybrid cross examples that Mendel did, was that he took seed color and seed shape and wanted to see what would happen. So he took a true breeding yellow round seed, which means that it had homozygous dominant for both of those characteristics, and a true breeding green wrinkled seed, which showed the recessive characteristics, meaning that they were both homozygous recessive. So here's Mendel's dihybrid cross, where he looked at two traits. The first trait looked at seed color. The second trait looked at seed shape. So he took true breeding parents, meaning they were homozygous dominant and then homozygous recessive. And what he ended up with was 100% offspring that were heterozygous dominant. And that's because the true breeding parents could only pass down either a dominant or a recessive trait. So the phenotypes of the F1 generation were 100% yellow and round. So now let's see what would happen if we wanted to take a cross of the F1 generation. And here's where we have the dihybrid cross, where along the sides we're going to write down the possible gametes. So first we've got our different traits. We've got our first trait for color, and then we've got our second trait for shape. So how are we going to figure out the possible gametes? Well, in order to figure out the possible gametes, let's explore what could possibly be passed down. So I could either get from here the passing down of a capital Y, capital R, so capital Y, capital R, or a capital Y, lowercase r. Or I could potentially get a lowercase y and a capital R, lowercase y and a capital R, or a lowercase y and a lowercase r. Now, do you notice how I always keep the y's first and the R's second. When you're doing dihybrid crosses, it's important to keep the same letter next to the other letter. Again, what are our possible gametes from our second organism? You get capital R, capital Y, capital Y, capital R, capital Y, lowercase r. I could get a lowercase y, a lowercase r, or I could get a lowercase y and a lowercase r. This kind of reminds me of like foiling in math. So now in order to fill out the boxes, all I have to do is combine our letters. And what do I mean by combine? So let's go through it. So here I would get capital Y, capital Y, capital R, capital R. Below it, I would get capital Y, capital Y, capital R, lowercase r. Capital Y, lowercase y, capital R, capital R, capital Y, lowercase y, capital Y, R, lowercase r. So take a second and fill out the rest of these crosses. So hopefully your Punnett square looks similar to mine, where what you ended up with was a 9:3 to 3 to 1 ratio. Meaning, if I have both traits that I'm looking at dominant, 9 out of the 16 possible offspring would express both dominant traits. If I were to look at 3 of those offspring, 3 out of the 16 would express 1 dominant and 1 recessive characteristic or trait that we were looking at. And if we were to look at all 16 offspring, 1 of those 16 offspring would express both recessive traits. Therefore, the ratio with 2 heterozygous parents in a dihybrid cross is always 9:3 to 3 to 1. So here's the picture form of what we just did, where you have a 9:3 to 3 to 1 ratio. So based on these results, Mendel formed 2 laws, the laws of independent assortment, where each characteristic is inherited independently, meaning that one trait does not determine the inheritance of another. So what would Mendel have gotten if the traits did not assort independently? He would have gotten that blending of traits. Therefore, this is further evidence that the blending hypothesis is not supported. The second law that Mendel didn't necessarily discover, but that his inheritance reflects the rules of this law, is the product law, is that the chance that 2 or more independent events will occur together in some combination is equal to the product of each event occurring independently. So if I were to break down that dihybrid example into 2 separate monohybrid examples, I would end up with the same results that I got doing the dihybrid cross. So here's my dihybrid cross broken down. I did a monohybrid cross for the chance of yellow and a monohybrid cross for the chance of wrinkled, and I ended up with the possibility of 3/16. Well, let's go back. Yellow wrinkled, was that 3/16? Yep, yellow and wrinkled. And guess what, guys? You can do the same thing for a trihybrid cross where you're using 3 traits. All right, so go ahead and practice some dihybrid crosses as well as the product law.